Hello everybody, my name is Emese Meglécz and I work at Exmeister University. Today I'm going to present you our pipeline VTAM, which is a robust pipeline for validating metabarcoding data using optimized parameters based on internal controls. It's no news for everybody here is that metabarcoding suffers from some pitfalls. For example, there are sequencing and PCR errors, but luckily they are fairly well controlled by intelligent clustering algorithms. Chimeras are also frequent, and we luckily also have the tools to deal with them. We can have highly spurious sequences, which is not easy to filter out during most of the filtering steps, but we can, based on taxonomic assignment, it's possible to pinpoint them. We have to deal with PCR heterogeneity, and with the use of different replicates, it's getting possible, but there are not many tools that actually deal with this kind of problem. Metabar is a recent air package, which seems to be a good tool. We also have to take into account tag jump and cross-contamination, contamination between samples. And this is really a problem because those are real biological sequences that should be filtered out from some of the samples. There are tools that are trying to do this, but unfortunately they are not very widely used. We also have to deal with pseudogens, especially for markers like CO1. And there are not very many, actually. I don't know any tools that filters them out explicitly. There have been many papers calling for good practice for experimental design, and this experimental design should go hand in hand with bioinformatic analysis. There should be biological or technical replicates in a data set, and it's becoming more frequent recently. The use of negative control is very important, and also there are more and more studies with mock communities, luckily. But what happens in most of the analysis is that those control samples are just treated as any other samples, and then after filtering, it will check if they comply to their expectations. If not, well, they have to go back to try analysis or other parameter settings till they find the results which suits to their expectation. This is a trial and error, and it takes quite a lot of time. What we would like to do in VTAM is to find a delicate balance between false positives and false negatives. And therefore, what is important is to find optimized parameters for each data set. But we cannot control all experimental fluctuations, experimental parameters and fluctuations. So each data set is unique, and the same parameter setting cannot really apply for each data set. So that's why optimization is necessary. So the VTAM pipeline will try to do something complete. We can start from paired and FASTQ files and end up with an ASV table with sequences assigned to taxa. I'm not going to throw all of this, but just want to point out that the filtering step actually is composed of several different steps, and each of them are aiming to eliminate some kind of experimental problems. The data set that enters to VTAM looks like this. So we have a sequencing run. So run is a sequencing run where we can have one or several markers. And if the markers are seriously overlapping, considerably overlapping, VTAM can handle them and pull the results of the two markers into one single table at the end. Obviously, we have different samples. Samples can be real samples, mock samples with known compositions, and negative controls. Each sample can have different replicates. Typically in the lab, we use 3 replicates, but it can be 2 or 4, depending on your financial resources, basically. So in VTAM, I've talked about very briefly filtering, and now I want to concentrate on the optimization step. As you see it here, from here, VTAM is not a linear pipeline. We ask users forced to filter out most of the errors by using the default values, which have very low stringency. So it eliminates a lot of errors, but not all of them. Users are asked to flag different occurrences in control samples. So we can speak about keep occurrences, which are expected variants in mock samples, and delete occurrences, which are all other occurrences in mock samples, and all occurrences in negative controls. What is happening in optimized step is that VTAM goes through a series of combination of parameter settings, and when it intends to retain all keep occurrences, so retain all expected occurrences in mock samples, while deleting or eliminating most of the delete occurrences. So by keeping all keep occurrences, we try to minimize false negatives, and deleting unwanted occurrences, we try to minimize false positives. And this optimization is done in filters, in in LFN filters. LFN is for low frequency noise filterings, which are actually very strongly influenced by experimental fluctuation, and that's why it's important to have an optimal parameter setting for each data set. We compared the results of VTAM to 2 other pipelines we constructed, actually 4 other pipelines we constructed. The first one is based on Dada2 and Lulu. Both of them are well-known tools. And because they don't deal explicitly with different replicates, we pull the replicates in the same manner as in VTAM, which means that we accept an occurrence in a sample if it's present in at least 2 replicates out of 3. The other basic pipeline we had was based on OBTools 3 and metavar R package, and we used the same kind of pooling of different replicates. Both of those simple pipelines were enhanced to create a pipeline as comparable as possible to VTAM. So one of the filter we added, which is a very simple filter, is the mean read count. So for each occurrences, if the number of reads is below a threshold, we just simply consider it irrelevant and we deleted the occurrence. However, the big problem is that we need a threshold, and neither the database nor the OBTool-based pipelines are giving an idea about this threshold. So in our test, we just simply took the value optimized by VTAM and used in the enhanced pipelines those values. We also added filters on pseudogens and also in filtered out chimeras, because they are not included in the original underlying tools. Let's see the results. We had 2 data sets from bat and fish excrements in both cases, and we used CO1 marker in both cases. So when you look at the precision based on control samples, so in control samples, we have the ground truth. We know exactly what it should be there. So we can calculate the precision. The precision is one of the highest in the VTAM data, in the VTAM filtering, and it's pretty low in the simple pipelines without the complementary filtering, whether it's based on Dada or Lulu, and whether it's based on the bad data set or the fish data set. Adding supplementary filters increase the precision considerably. However, as I told you earlier, this supplementary filtering was based on a on a parameter value optimized by VTAM, and usually users don't have this kind of information beforehand. So precision is pretty high in VTAM, and in sensitivity at the same time is comparable among all pipelines. Actually, the basic data pipeline is performing better than the other one in sensitivity, but just very slightly. When we compare real samples, so he eliminated all control samples and we had just real samples here. We calculated ASV richness, so the number of variants in each sample after different filtering, and the ASV richness was lowest in VTAM significantly compared to all of the 4 other pipelines. And it was true for bats and fish. So what I'm trying to say is whether how we filter the data has got quite a lot of influence on ecological consideration. If you want to estimate the richness, it does matter what are the parameters we are using and what kind of pipeline we are using. Species richness or ASV richness can be significantly overestimated if we don't filter the data with sufficiently stringent parameters. And at the same time, as I pointed out here, filtering efficiently parameters really does depend on parameter values, which should be optimized previously, and this is done by VTAM. At last, I would like really thank all my colleagues who took a great part of this work, and thank you very much for your attention.